Good afternoon. You guys, it's amazing you're here after lunch. You know, lunch is a sleeping pill, right? So don't go to sleep while I talk. All right. Oh, that works. Okay. I work at FHP, basically seeing cancer patients and people with blood problems. Everybody is talking about screening. You all know about screening. You hear about it all the time. So I thought I may tell you a little bit about cancer itself, why we need to screen. And I think once you know a little bit about that, I guess half of you or more than half of you already know it. But if you don't, maybe this will help you a little bit to figure out. Of course, we even haven't figured out, but I can give you some basics that we know, which is people that know way beyond this, that do that, they can tell you how important it is to find out what causes cancer and how it happens. So that comes out to be why the screening is needed, because if you understand a little bit about the growth of cancer, you'll understand why screening is needed. Next slide. So what causes cancer? I think you hear about it every day. This thing causes cancer, this thing causes cancer. Actually, we really don't know, okay? I think there are multiple reasons for it. There's not one reason for it. We know just a few. We just know just a few that we can elaborate, and I'll just mention whatever there is. Next. So I put that at number one on my list, ionizing radiation, because this happens to everybody, you know? Ultraviolet rays, being in the sun, especially in Guam, other islands. But luckily, most of the people in Guam and other islands are brown, so their skin helps them out with melanoma and other skin cancers, but people with lighter skin are really at very high risk in these areas, in all these islands. And I've seen most of the melanoma patients I've seen have been in people who have origins from US or from Europe, and I haven't seen one melanoma yet. I've been here 3 years, not one melanoma amongst the locals. But they get a lot of other skin cancer, like squamous cell cancers, but no melanoma. Next one is the exposure to radiation that we get on cancer treatments, or otherwise, like exposure to radiation like it happened in Japan, you know, that kind of exposure. When you get exposed to radiation, it does something to your cells in the body. Whatever happens to the blood cells, basically it mutates and ultimately turns into cancer. So any area in the body where the radiation goes through, and Dr. already explained quite a bit of it, like somebody getting radiation to the lung, sometimes it has to go through the breast, and a part of breast get exposed, so you can easily get breast cancer. Some of the lungs get exposed, you can get lung cancer. So the risk is there. Of course, not everybody gets that, but there's a risk, there's a higher risk. And people who were treated for like Hodgkin's disease, lymphomas, 20, 30 years ago, thought they were cured. Some of these people, about 10%, came back with different cancers in their body. Like wherever the radiation was given, those areas got involved. So that's a cancer risk. Although Dr. Rao can cure a lot of cancers with radiation, but at the same time, radiation can also cause cancer. Chemicals, especially everybody knows about cigarettes. Pesticides, I think, is one of the major reasons nowadays. And really, we know only about animal data. We don't know much about human data on pesticides. But I don't know of any food that's made out there does not use pesticide. Because all the vegetables, all the fruit, everything, they use that to make it big. Beetle nut here, and Dr. already mentioned that earlier, industrial exposure, asbestos, auto emissions. Now, I wouldn't have thought of auto emission has been really one of the major reasons for the lung cancer, along with smoking. But I was reading a book that was published in 1896, and they were basically talking about growths. Of course, they didn't know much about cancer. They knew they were growths. They knew some growths are abnormal. And they were talking about these growths, quote unquote, cancers in autopsies in different people. And interestingly, they found lung cancer was at the bottom of the list. All the other cancers were much commoner than lung cancer. So what happened between them? They used to smoke just the same tobacco everybody else smokes. So what is in cigarettes? So cigarettes really is not 100% tobacco, as everybody knows that. Tobacco doesn't smell that good, doesn't taste that good. So if you ever had real tobacco, you can tell you can't really stand it unless you're really into it. But apparently, they're not treated with any chemicals. So your exposure to chemicals is much higher because of the treatment of the cigarettes. Viral infection, and Dr. already mentioned about HPV, and also, you know, what head and neck cancer, cervical cancer, and getting vaccination becomes an important part. And again, same thing like EB virus. And then Helicobacter pylori bacteria in the stomach, which is found in the ulcers, can develop into malignancy, can develop cancer. So bacteria, and there are other bacteria in the body doing that. Some of them we know, some of them we don't know. There's a lot to be learned about cancer origins and correlation with these different organisms in our environment. Hereditary exposure, for example, breast cancer, and something we are finding out now, because if you guys ever heard about next generation sequencing, if you haven't, what it means is basically you can take a DNA from inside the cell, and then you can take one particular gene and find out that particular gene, how many abnormalities exist in that. So they found out that certain cancers really don't fit in what we know. We know about 2 genes right now, BRCA1, BRCA2, that are associated with breast cancer. But they are finding out there are about 10 different genes, actually, that are involved with this. And there's a lot of people, especially in this area, in Guam, and from Hawaii, and from Manila, that are coming in with hereditary familial cancer, running in the families, and they have gene mutation in the germline cell, and they are not BRCA1 or BRCA2. I've seen only maybe one patient with BRCA1 and 2 positive. Everybody else tells me my mom has it, my aunt has it, my grandma had it, but BRCA1 and BRCA2 is negative. So what's going on? Are we really finding everybody? Probably not. So next generation sequencing, when it's complete, the data is being compiled. Probably in about 5 years, you're going to find out a lot more genes than you know, a lot more genes, and that's coming up very soon. Lynch syndrome, have you heard of that? No. Lynch syndrome is known in malignancies, especially in colon cancer. And we found the gene that causes, basically it causes multiple malignancies, including colon cancer. So people who have colon cancer, you can do Lynch syndrome study and the gene for that particular gene involved, because the family, colon cancer also runs in the family, just like breast cancer does, same way prostate cancer. Obesity. So what you got exposed, what you inhale, what you eat, basically you're inhaling cancer-causing agents. Big risk factor. So that's why I think Europeans and a lot of people in USA, especially colon cancer is getting so high. You know, you got obesity, you got a lot of food intake, breast cancer, higher risk. Those 2 big cancers we already know associated with obesity. All the food stored in grocery stores and chemicals are processed. Majority of that is processed food, chemicalated. So, anyways, you can't get away from it. Next. So how does it begin? This is how it starts. Here's a normal cell, and here's the chromosomes. Chromosomes basically are found inside the nucleus, inside the cell. Here's the cell. That's how a normal cell look like. So if you put a bunch of cells together, they're like little bitty tennis balls, very, very minute. You can only see under the microscope. When you put them together, they make a solid organ. So like our stomach and our heart and our colon, our breast is made up of different cells. So we can take any cell that's normal. It has these chromosomes in them. The chromosomes, basically each one of these chromosomes, there are 23 chromosomes. Each one of these chromosomes, 23 pairs. Each one of these chromosomes represent a gene. Represent a gene that particularly represents a certain growth that's going to happen, certain signals that are going to go through to that organ to grow or not to grow. But something happens to one of these genes, okay? It's called mutation. This is where the problem begins. That's where all these chemicals come in, radiation come in, chemotherapy comes in. Whatever exposure you have with chemicals, this one mutation in one gene starts it. But we don't know which gene, and we don't know what time or when it happens, but we know it happens because we've seen that in animal models and also humans. One mutation means a change in the chromosome, okay? So this particular gene inside the chromosome, because there are thousands of genes in each chromosome, one gene is mutated, mutation means abnormality or abnormal, can lead to 2. Then it leads to 3. Look at the cell. How is it deforming? See the color change? The real cell doesn't change color. It just changes the shape of the real cell. The real cell now, which was normal cell one time, after this gene mutation begins to show some abnormalities. These abnormalities lead into a weird-looking cell called cancer cell. It's interesting, the cancer cell look different than normal cell, bigger. So here's normal cell, very nice, round. Look at this malignant cell. So how long does it take from here to here? Several years. As far as we know, the exposure may have happened 10 years ago, like smokers may have smoked like for 20 years, quit. Then all of a sudden, 10, 20 years later, come up with lung cancer. And you say, wait a minute, they're saying I haven't smoked any for 20 years, but that exposure right here already, gene mutation already began. When that began, that led to cancer that showed up later. So it happens all the times. Happens with breast cancer. All the cancers actually start somewhere, from one cell to multiple cells. Now this cell here has a lifespan, okay? A normal cell in our body in different organs live different life. Okay, like for example, in the blood, we got blood cells, like red blood cell, platelets, white blood cell. They have a life. All cells in our body have a life. Some have 3 days, some have 5 days, some 7 days, some 100 days, but there's a life. Just like humans have life, same way our cells have life, they die. But then they regenerate again, the new ones come in, but they die. So something is killing them, right? So when they die, something is telling these cells to die. So this happens from signals. The signals start right here on the surface, telling the cancer cells to live. After 3 days, you're going to die. Something else comes up there and gives the signal, die. And the cancer cell or normal cells die. But these cells, cancer cells, don't know how to die. They are told, don't die. That little gene that tells you die is not there anymore. It's been mutated. It's abnormal. So in reality, after that many mutations, that gene that stops you from growing, not there anymore. So the cancer cells just keep on dividing, and doesn't know when to stop, because it keeps on getting those growth signals. So in reality, it really happens, because a lot of experiments have been done, and it has been shown that you can actually put the growth factors in there and make things grow. And we do that all the time in our clinic in oncology. We give these growth factors and make the white cells grow. Because we can stimulate certain areas that could start producing more. But these cells here, they grow, but they don't know where to stop. Anyways, next slide. This is real. This is an electron microscope of a normal cell. See these little hair? Little hair are receptors. Receptor means they receive signals. Okay? So there's so many receptors, look on the cell, and they represent different things being told to the cell, like produce insulin, or produce thyroid hormone, or whatever. You know, it's getting signals from outside. There's the actual cell. Inside the cell is the nucleus. This growth signal goes to the nucleus, and they tell the nucleus to divide. So this is how the normal cell is, but look at this one here. This is transformed, this mutated cancer cell, in reality, this is how it looks like under electron microscope. See these here? They look like ninjas. They look crazy. This is what attacks you, because there was only like maybe one or two these little receptors representing something. The same receptor may turn into like 20 different receptors. Same kind. So if you look at it carefully, you find these little 2 eyes and a nose and a mouth. That makes it a devil. So I call it the cancer cells are devil cells. I'm making it up. Next one. I don't want to confuse you with all this, but this is what goes on in your body. Normal cells. See these receptors I showed you earlier? These are the receptors, taking in the signals for growth. But look at this. Here is what we call the death receptors coming in through here. This end. This is what causes the cancer right here, called apoptosis, makes the cancer to die, or any cell to die. So if you have a problem with these areas here, the cancer cell keep getting signals from here. All these other little electrons telling you, go, grow, grow, grow. Next. So what happens in reality is there's an actual gene, and we found out what that gene is. This gene causes a normal inhibition. It stops the growth of it, okay? So the cell division is under control. It would divide 20 times, 30 times, and die, right? Here is the defect in the same gene, and we know what those defects are too. So this mutated gene has the defective protein, no stoppage. Look, here were only 2. By this time, the 2 are here, cancer cells are 20, then 30, then 1 million. It goes on and on and on. Next. This is the real picture of cancer cells. That's how they look like. All look the same. This is interesting. That's the funny part, is all look alike. There's nothing else in there. Then the cancer cells make their own blood vessels. And I think Dr. Raw told you about angiogenesis. Means the cancer cells have the capacity to make their own blood vessels. That's the dangerous part in cancer. All cancer cells do that. When they do that, that means that they can make little ways to get out. So if you got a blood vessel sitting somewhere, that you can easily transport yourself, the cells can start going into the blood vessel and spreading, right? Okay, next. For breast cancer to be visible on a mammogram, it has to be more than 0.4 centimeter in diameter. Okay? So anything smaller than 0.4 centimeter does not show up on X-rays. Now that's really scary, isn't it? So you go out there and you think that you got it. Your mammogram found it, or a CT scan found it, but you're really already late. Already late. Here is a cancer cell. This is not visible on mammogram, the cancer cell, but is already growing and spreading, okay? But is not visible on mammogram or chest X-ray or CT scan. That's the problem with cancer today. So by the time it's 0.4 centimeter, it's already grown a lot. It's got up close to 1 billion cells in it. And those cells, all they do is spread. So what do? They spread into blood vessels, lymph vessels. And that's what's called metastatic cancer, spreading all over the body. So in reality, the tumor grows from a single cancer cells, grows into this, what you see, then grows into this, then gets bigger. So where do we find it on a mammogram? Right here. So what happens between here and here? Spreading. So in reality, by the time we diagnose cancer, it's already spreading. So why is that? If you take it out right here, you can cure it sometimes. Why you can cure it is because our body takes care of those cancer cells that escaped. Our body has an immune system to kill those cells. Body does not like those cells and it kills it. But when it gets to a certain point where too many cells escape, the body doesn't have any chance to kill it. Or your body immune system is not good, has no chance to kill it. So whatever happens, our body is unable to take care of those escaped cancer cells, trouble begins because they're already gone out of control. So by the time it's visible to us, it's already in the liver, lung, everywhere, just sitting there. That's why nowadays, even very, very small cancers that the surgeon says, I took it out, I think you're cured, about half the time they're right, half the time they're wrong. Because the other half time they're wrong, it's already spread. And the half time they're wrong, they're right is their patient's body took care of it. So that's why we treat these people with combined treatments, chemotherapy, radiation, surgery, so we can knock out the escaped cells. Next. They invade the neighboring tissue. So it's invasion, it's a real invasion. Matter of fact, they go on almost like a, what can I say, little crabs just dig. The crab dig into the sand. This is exactly those little claws go out and they dig into the other tissues to eat through it. That's why a lot of times you see cancer eating through the skin and in the breast and it's showing up on the skin. And you see in a picture Dr. Oz showed about the cancer in the mouth that grew through the skin. Next. They spread through lymph nodes and blood vessels. Next. So, out of these cells that escaped, a small percentage will sit there if they are not treated. Or even if they are treated, they don't shrink or they don't die. So what happens? They make another little cancer, just like the one you started off with. So if you start in the lung, the little part that showed up in the liver would look just like lung cancer. If that part shows up in the bone, it would still look like lung cancer, although it's in the bone. So it's like seeds, you know, you spread a seed of a certain tree, the seed is going to grow into that particular tree. It's the same way as the cancer. Next. So what we are seeing on X-rays is right here, guys. Tip of the iceberg. Anybody seen an iceberg? Not in Guam, right? Okay, I went to South Pole last year, and I really, for the first time ever, saw an iceberg. I did not know what they meant by this. This is real, because you could see just maybe 5 foot big, and you say, oh, no problem, I can take my boat right there. No. Stay away from that, because that's not just 5 foot, it's like 100 foot below that is under the water, because the other 5 foot just couldn't go down deep enough, because as you know, snow and ice is a little lighter than the actual water is. So this is a real iceberg, and our cancer represents what we see on a radiology, X-ray, and mammogram is tip of the iceberg. This is what actually has already happened to the cancer. Next. Same thing what I told you earlier. Next. Same thing I told you earlier. So basically, if the cancer is here, you can imagine it going right here. You can imagine it going all over the body. because the circulation is all over the body. Certain areas on the body have more food than the others, like brain, lung, liver, bones. They have a lot of food. So the cancer cells love that area. That's why I see most of the cancer in the areas that I just mentioned. You don't see many cancers show up in the fingers. Has anybody seen a cancer metastasize to finger? Rarely, hardly ever. You see in the skin. Skin has a lot of blood in it. So wherever there's food, the cancer cells can live and survive. Next. So, smaller the cancer, higher the chance of cure. Is that a correct statement? 50% of the times is correct. But the other 50%, you have to find out where else is it. And some cancers, we do know that just the surgery or radiation is not going to cure them. They are going to need systemic treatments like chemotherapy to get rid of all the other cells in the body. Next. So, how do we detect these really, really small cancers? Dr. Hall is going to mention something about mammograms. That's one way. Things are changing, and I think we are going beyond regular technology of X-rays. I think what's going to happen is we're going to find into nuclear images, we'll find a gene ultimately that's abnormal. We're able to hook it up to something, tag it up, and show it just like the PET scans, PET scan. You can see tumors because you hook them up to glucose. Wherever the glucose metabolism is going on, you see cancer already on the plain films. It's the PET scan only detect a centimeter tumor or bigger. Smaller tumors doesn't show up on the PET scan. That's why we want to develop a PET scan that can show up those less than 1 centimeter tumors. Once we get there, I think we'll be able to definitely cure this cancer. Next. So, Dr. Hall has a whole series of controversies. So why do we do screening? Millions of dollars are being spent on cancer screening. I found our American Cancer Society's business is $1 billion business. Out of that $1 billion, only $400 million goes towards all the stuff the American Cancer Society does. $600 million are spent on other things, like people have to be paid to run these marathons. I mean, somebody has to do it, organizers, and there's a lot of thousands and thousands of people living off of American Cancer Society or other organizations like that. So in reality, screening is not cheap, it's expensive. Actually, there's more money needed to spend on cancer prevention. That's where the emphasis should be. Prevent it before it starts. That's why I was showing you all those divisions of cancer cells to say, stop it when the first attack happens or before the first attack happens. Stay away from all those bad things that can cause that particular cancer. We have found actually that 20% reduction in breast cancer mortality, and that's a real figure. Is that figure really true? That's all about screening? Maybe not. I think some of that is because of new technology with the treatments. We have a lot of new treatments. We are finding out like a gene called HER gene, HER, HER gene, we can attack it with Herceptin, which is an antibody against HER gene. We found out another drug called pertuzumab that can also attack. We have found 2 or 3 other drugs that can actually attack the gene and kill the cancer cells. So in reality, there are more ways of getting rid of cancer today than there were 10 years ago. So our data on screening is old. Our data on finding out what is really helping today's cancer, is it really screening or is it really new treatments? Unless we find that out, we don't know where the money is supposed to be spent. High expense of false positives. Mammogram, and Dr. Hall will elaborate, has so many false positives. So do you know how many biopsies are done of false positives? Thousands of biopsies are being done of false positives, which are nothing. Just a little bump in there, and mammogram shows up, and everybody's running for biopsy. That's money, that's cost. So it goes to healthcare. There's no benefit found on prostate screening. You know how much money was being spent? Millions to billions of dollars was being spent on prostate cancer screening until somebody did study over 10, 15 years and found out that really a lot of that was a waste of time in biopsies and early treatment when the prostate cancer patients lived 10 to 20 years without treatment. Without treatment. So that means that really the screening didn't help much to those patients. So as far as we know, prostate screening is now done only by informing and talking to the patient and asking them and seeing the risk factors. Not every person should get routine screening done. But talk to the person or talk to the patient and see what else he's got. Heart disease, lung disease, something else. Some people are going to die like in 2 years or 3 years from something else. Are you going to do all the screenings on them? I mean, they're going to die from lung disease, heart disease. So we have to really be very prudent in talking to our patients and looking at them and say, what's the life of this patient? Like a prostate cancer screening on somebody who's 80-year-old, is it really worth it? That guy is going to make it even with the cancer for 10, 20 more years. So why are we doing that? Why are we biopsying him, and why are we trying to treat him and putting him through misery? So we have to really look carefully now on screening and how much money we want to spend and how much needs to be done. Discussion definitely needed between the patient and physician. Certain areas are very important, and I think we do know the Chamorro population and other island population, native population in Guam is a very high risk. I think those are the people we know, because they do have a higher risk of cancer. We do know that education and screening is needed, definitely, regardless of the data. So yes, we should continue on this kind of population, but when you know a regular place like New York or somewhere else where most of the people know what they're doing, they get all the regular screenings done, that they're not at high risk, they don't hide their bumps, you know, they get pelvic exams done regularly, you know their risk is lower in those people. So screening should be based upon risk factors in the person, should be based upon what's the population like. So Guam population, especially Chamorro population, definitely needs screening, more education to know which way we are going and where the money should be spent. Next. Thank you.
All right. Thank you one more time. Please give it up for Dr. Chaudhry, ladies and gentlemen, from the FHP Cancer Center.